Okay. Hello everyone. Good evening everyone. Welcome you all to our YouTube channel AgriAddict. So I'm back with the second part of the transmission genetics. Yesterday, even though I did not commit that I'll come up with another session, but since because of the kind of response that you people have provided for the previous video, I tried my best to come up with a second one. Like I have prepared the rest of the content for transmission genetics so that the video should feel complete. And once you watch the video of Mendelian genetics, all should be explained. Okay. So please confirm once, like if I'm audible to all of you, and very good evening everyone for each one of you. Okay, thank you so much. So I'm audible. You have joined. Please share the link with all your friends, like who are preparing for this exams, at least for your friends who are doing the self study, who are doing the group study, everything, so that can later discuss these questions, like later discuss the topics, the concepts that I am going to discuss in this video. Fine. uh, how about the yesterday session? Like how many of you felt it like, okay, we have covered something which is very big in a matter of just 1 or 2 hours? Was it really useful? What do you think? So what was left from the previous session? Yes, okay, like some students are requesting for the molecular genetics. Yes, I will come up with the molecular genetics, do not worry about it. So it will be mostly by tomorrow evening or latest by Monday morning, for sure. That's my promise. But I have one request for you, like if you want me to come up with a video, I want you to comment on this video once the live streaming is ended. Please put your views, put your comments, show some love, support, so that we will be able to reach more and more students and build this platform and help more students like you, so that students can get a better rank, a best rank in their ICAR PG exams in the coming years too. And I hope like you might be knowing about the upcoming online courses for the 2026 and 2027. The way you have enjoyed this year particular course, let your juniors also enjoy the same learning. Let us make the learning enjoyable. So please let them know about our courses so that they can enroll in our courses and they can also cover the syllabus and get the best rank in the upcoming exams. Okay, it's my sincere request that if you have got some value out of our online courses, I would request you just share this with your juniors and let them come to the platform, let them know the platform, introduce all of them to the platform so that they will be also benefited the way you are getting benefited. I hope like you people are getting benefited from the courses that we have launched for this year, and we had created some good impact in your preparation. Okay, let's start. Fine. What were the topics that we did not discuss in the previous class with respect to the Mendelian genetics? What topics we did not discuss? The mutation, okay, multiple alleles. Yeah, the two topics that I left. So now I will be again discussing, I will be again starting with the cytoplasmic inheritance, because in the yesterday's marathon series, I was not completely comfortable the way I discussed the cytoplasmic inheritance. So I'll be again starting with the cytoplasmic inheritance. I'll be discussing only two topics today. The video will not be too long since it's almost like 10:30. So I'll be taking not much time, but I'll be discussing each and everything in detail so that it should be easy for you to revise. So two things I'll be discussing in today's video. One is regarding the multiple alleles, and the second one is regarding now the cytoplasmic inheritance. Students, you asked about the mutation. The mutation transposable elements will be discussed along with the molecular genetics. Okay. Once the molecular genetics is done, if you want me to discuss the other part, which is like a general history plus the cytogenetics, I will come up with that video by hopefully by Tuesday. Okay. Not if not Monday, I'll come up with that Tuesday after completing the molecular genetics. Plus, the students have also asked for the plant pathology marathon series. So I have already requested Parul ma'am to take these classes. She has agreed to take the classes, but it can't be live since she's not at Delhi. She's in our college PAU. So she will be recording the class and she will be uploading it on the YouTube directly. So Once it is uploaded, please make sure to watch the video and take a full benefit. And this particular marathon series, it is for you people who are going to write the exam on the July 3rd. So it's your responsibility that you have to go and check the video and watch it completely and take the complete benefit out of the course. See, whatever you have requested, we are coming up with this. We are making our best efforts to make sure that whatever you ask for, we will deliver it. Okay, fine. So let's begin the today's class. So today I'll be starting up with the cytoplasmic inheritance. So basically, before going directly into the cytoplasmic inheritance, let's discuss about two different categories. See, one is called as Mendelian inheritance, other one is like non-Mendelian inheritance. So what is the difference? See, if you know from the Mendel's first and second law, you know the contribution of will be from the both male and the female from generation to generation. The alleles, the chromosomes will be contributed, or the traits will be contributed by both males and females, and it is like equal contribution will be from both the males and females. That is the very first point that you have to remember. This is under the Mendelian inheritance. Then the reciprocal crosses are always equal. So what I mean to say, the reciprocal crosses, if I take an example here, let's take a seed color. Let's assume a seed color which I'm talking about. If for example I'm talking about red is crossed with white, where red is from the female and white is from the male, and the other the reciprocal cross of it will be like white will be female and the red will be male. So the result of these two crosses in the both the cases, it will be red, since if it is under the Mendelian inheritance or if it is following the Mendelian inheritance. That's what the reciprocal crosses are equal means. Next, it shows the typical 3:1 ratio and 9:3:1 ratio in case of F2. If this is the F1, if this is self, this will show a typical F2 ratio, which will be like 3:1 in the phenotypic segregation, and if it is bound by the monogenic, and if it is dihybrid cross, then it will be like 3:1:1:1. Okay, so this is what we have to remember. So this is about the shows typical 3:1 and 9:3:3:1 ratio. Next, very importantly, the genes or the traits are the traits which are showing this behavior, which showing that equal contribution from male and female, showing the reciprocal crosses are equal. So they are governed by which genes? They are governed by nucleages. So these are the very important points that you have to remember. So whatever deviation that we see from these particular characters, those are considered under a category called as non-Mendelian inheritance. Now tell me, if I say non-Mendelian inheritance, is it a cytoplasmic inheritance which I am talking about? Is it the cytoplasmic inheritance which I am talking about, or it is something else? So what do you think? Quick. Open the chat box and keep. Okay, it's a two-way learning, not a one-way learning. Okay, you have to interact with me. So both cyto and nuclear, not only nuclear. Okay, students are getting my point. So this is the place where most of us will not have the clarity. Okay, let me clarify this. Whenever we are talking about the Mendelian inheritance, if it is non-Mendelian inheritance, it does not always mean that we are talking about cytoplasmic inheritance. See, first of all, define cytoplasmic inheritance? The cytoplasmic inheritance is an inheritance pattern which is because of cytoplasmic genes. Okay, and whatever deviations that we are seeing here, like generally by the females, reciprocal are different, and there is no segregation in the F2 or in the subsequent generation, and this is not like this is by not by this is like not by nuclear genes. Okay, add it here, it is not by the nuclear genes. So these characters are not limited to the cytoplasmic genes. Okay, so there are also other examples where the non-Mendelian inheritance will take place. What are the examples? I'll come up with that. So before that, what is the cytoplasmic genes? So where are these genes located? The cytoplasmic genes are located in the two important cell organelles, which is like mitochondria, first one, and the second one is chloroplast. And the DNA which is present, in which like you can find the genes, the information, they are called as mtDNA, mitochondrial DNA, and the cytoplasmic DNA. So in the simple words, if I talk about the cytoplasmic inheritance, that means the genes are located only in CP or mitochondria. Only two cases are there for the cytoplasmic inheritance. So okay, I have two different terms again. One is like maternal effect and the maternal inheritance. So tell me whether these two are same phenomena or there is a difference phenomena. Yes, same or different. What is it? Yes, quick. Maternal effect and the maternal inheritance. Are they same or different? So please remember these two, although seems similar, but they are different phenomena. Very good. Who is answering it? Okay. So here maternal effect. What is the clear cut difference between the maternal effect and the maternal inheritance? So maternal inheritance is the another name for cytoplasmic inheritance. That means here in this case, if I have to differentiate, what is the main difference? Can you just anyone tell me like what is the main difference between the maternal inheritance and the maternal effect? Yes, please. जल्दी जल्दी बताओ. So what is the main difference between the maternal effect and the maternal inheritance? Maternal effect is by the nuclear genes. Okay, so the maternal effect is by the nuclear genes. Good. That means the whatever effect we are seeing in whichever trait, the trait is actually governed by the nuclear genes itself. Okay, there will be influence of the genotype of the female. I'll come to that later. But in the first place, this is because of the nuclear genes only. But whenever I talk about the maternal inheritance, which is the other name for the cytoplasmic inheritance or extra chromosomal inheritance, so this is governed by what genes? This is governed by the cytoplasmic genes. If I say that cytoplasmic genes, what does it directly mean? It directly means that genes are either present in the mitochondria or the chloroplast. This is the basics about the cytoplasmic inheritance. Okay. Now, let's talk about non-Mendelian inheritance. In the non-Mendelian inheritance, I told you about one clear-cut class that was regarding the third one, which is cytoplasmic inheritance, where it is governed by the DNA, which is present in the CP and the mitochondria. Okay, the last one is called as maternal effect. So this is also not showing the Mendelian inheritance pattern, but it is showing the non-Mendelian inheritance. How? Even in this, the first case, the reciprocal crosses are different. It is not same in the maternal effect, and here it is also different. Or overall, if I talk about here the first case, the reciprocal crosses, if I talk about, are different, and there is like not equal contribution will not be there here. We won't see such pattern, and there is no segregation. I'll tell you like what is segregation in the upcoming slide. No segregation in F2. So that we observe in all of these patterns, but these patterns are actually different from each other. That's what I'm trying to say. One clear cut pattern is like cytoplasmic inheritance. We know it. This is because of the genes present in the chloroplast and the mitochondria. The other one, although like genes are present from the nucleus, but still it shows some difference from the Mendelian inheritance. That's why we are kept it under non-Mendelian part. Okay, the first one, it is related to the cellular structures and the patterns, and second one, it might be because of the intracellular parasites and the symbionts. So can you give me the example for all of this here, starting one by one? So what are the examples? Examples are the most important. Remember, in this chapter, you have to remember the examples for all the different categories. Can you tell me the examples here, starting with the cellular structures? These are mainly related to the bacteria cell wall. So this is related to the bacterial cell wall. Although like the entire mechanism is not clear, so need not to worry about it. Just remember that it shows the non-Mendelian inheritance, but here the genes are actually from the nucleus only. Okay, there is no change. Next, they're produced by the intracellular parasites, symbionts, and the virus. As soon as you see the term parasites, what is the one examples that you get in mind? What is the example for this particular category? Yes, quick. Anyone? The examples I told you, like it's very important. And one particular peculiar example is, yes, very good. It is kappa particle. Good. Students are answering it. So kappa particles. So now don't scratch your head. Do not scratch your head to understand like what are these kappa particles, what is the inheritance pattern. So if you are a student of master course, you must be already knowing it because I have clearly explained it in the class. But if you are doing your self study, if you don't understand it by any chance, do not worry about it right now. Just know the example that this is the example for the non-Mendelian inheritance. It is like kappa particles in which organism? It is in the paramecium. Okay, that's it. That's it. Okay, next thing. DNA containing cell organelles. I told you it is in two different cell organelles, which is CP and MT. So what do we call the DNA that is present in the CP, and what do we call the DNA that is present in the MT? I told you yesterday, if you remember. This is called as chondriome and it is called as plastome, right? And what do we call the overall genes present in cytoplasm? What do we call that? What do we call this? Yes, I want the answer. What do you call for all the overall genes that is present in the, yes, it is plasmon. So try to remember the terms here. Do not get confused with the plastone and the plasmon. Okay. Now, next, what else? Examples. One thing is like I always go with the examples and I will always ask the example. So what is the examples for both of these two cases? Tell me the examples. One very peculiar example here, it is like variegated leaves. And if you know, the cytoplasmic inheritance was for the first time discovered in Mirabilis jalapa. Who was it? Who for the first time discovered the phenomena of the cytoplasmic inheritance? It was by Correns, right? It was in the Mirabilis jalapa. Okay, and the peculiar trait was the variegated leaves, where the leaves were producing like white, like they are producing the green and the variegated. So what is this variegated? What does it mean? It will have the leaves where the white patches will be present on the green background. That's what the variegation means. And this particular trait was showing the non-Mendelian inheritance, which is because of the reason that, so this trait is governed by the genes which are present in the chloroplast. Okay. The second example. So what are the examples for the mitochondrial DNA? So mitochondrial genes. So what are the examples? You should be able to say this. The examples for this is, one is Petite mutant. If you know Petite mutant in which organism, it is in the yeast. And the second example, it's a very well known example, which is the CMS, cytoplasmic male sterility in case of plants. And who discovered this cytoplasmic male sterility in the plants? Who described it? So it was like, there are also other example, LHON. Yes, I agree with you, but these two are minimum required, which are needed to be remembered. So you remember this, and if possible, remember all the other examples. Even I have given the all list of examples that are available for the mitochondrial DNA or the mitochondrial transmission, right? Good. So if you know the things, it's well and good. Know it. Okay. Even this can be asked. It doesn't mean that it won't be asked in the exams, but I am just focusing on the very important content. Okay. The pattern mutant, it is in the yeast, and the CMS in the plants. So who described this for the first time? Who did it? Yes, it is by Rhodes, if you remember. Okay. And what is the CMS? If I have to explain, I'll do it. So next one is the maternal effect. So what is the peculiar example for the maternal effect? Who did it? Sorry, what is a peculiar example? Yes, 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 quick. Examples for the one good example of a trait in case of snail. Yes, it is like shell coiling in the snail. So good, like people are knowing the examples. The snail shell coiling. Okay, see, be confident. If you are able to answer the questions that I am asking in this marathon series, consider that you have prepared most of it for the ICAR PG exam, because the difficulty level of the paper will not be that much high. If you know these minimal things, you can apply your mind and you can answer it. Be confident about it, and I'm feeling very happy that you people are able to answer this. This is the shell coiling in snail, where the two different traits that are available here, it is dextral and sinistral, right hand coiling and left hand coiling. Okay, in detail, if you know it about like what is the difference between the like, see, you know the difference between the reciprocal process here. In this case also you find the difference. Yeah, so this is I have mentioned about, I'll come to this later. So I'll just show you about the maternal inheritance. So here, if you know, this is dextral and sinistral. This is considered the trait of for a character which is snail shell coiling. Okay, this is the trait. Two classes. If the female is dextral and the male is sinistral, then the progeny which we are getting, it is dextral. Okay, so if this is gone by a single gene, one expectation that we can keep is, because the dextral might be dominant, and that's why we are getting dominant in case of that's why we are getting dextral in case of F1. But when the reciprocal cross is done, you can see here the male has been used as a dextral and the female is used as a sinistral. But in this case, we should again get dextral if it was according to the Mendelian inheritance. But here we are getting sinistral. So one common thing, what it is like whatever the female phenotype is there, it is directly getting inherited. That's what the maternal effect is. But one thing you should not miss here, that is you should not see the phenotype of the female but the genotype of the female. So here it's not the phenotype of the female is transferring to the next generation, it is the effect of the genotype of the female parent which is shown in the progeny generation. As you can see, if there is a DD which will definitely gives the dextral, then the progeny will be automatically dextral irrespective of whether it is sinistral or dextral in the male. Okay, similarly if this is crossed, it is selfed. Okay, if this is self, irrespective of whatever it is, like all of them will be dextral. Okay, the next generation in the F3 generation, if this is self, okay, this will be dextral for no doubt, this is self, this will be dextral for no doubt, and the third part again it will be dextral for no doubt. But this one, even the phenotype is dextral, but because the genotype is like small d, the next generation will be the sinistral. So this is what exactly the maternal effect means. If in the exam they are asking you in case of assertion reasoning or in the statement wise questions anything related to the maternal effect, its definition, its example or the mechanisms, simply remember that the progeny trait, progeny trait is directly dependent on the genotype of the female parent. Which parent? It is from the female parent. This is everything about the shell coiling that you have to remember, nothing else. Okay, next we'll jump into the exact cytoplasmic inheritance in bit detail. First thing, very important is to remember the characteristics of the cytoplasmic inheritance. First thing, you see the reciprocal differences that you also seen in the maternal effect. Then there is a lack of segregation, but this is a difference. This is a one thing which you can differentiate between the maternal effect and the maternal inheritance. Now tell me whether even this is true in the maternal effect, that there is a lack of segregation. Don't you see the segregation in case of maternal effect, the traits which are showing the maternal effect? Is it true? Yes or no? Tell me whether the traits which are showing the maternal effect are giving the segregation or not. There is a segregation in those traits for those traits or not. Okay, one said no, segregation in the F3. Yes, there is a segregation. Please remember, do not think that there is no segregation. See, no segregation means, in a simple words, what does it mean? That means in all the generations, the n number of generation we are going, should get only one of the true trait. That's it in the population. But here up to the F2, it was dextral. But as soon as we went for F3, the sinistral also came, where there was a segregation for this trait, particular trait, where both like the one which was missing in the earlier generation, it reappeared. So in this case, remember maternal effect is not the complete lack of segregation, but it is like segregation won't occur in F2, it usually occurs in the F3. And I told you like what is segregation, right? Segregation is nothing but the all the traits should be expressed in a population. No, nothing should be missed. If only one trait is expressing, that means there is a lack of segregation, and it is mostly because of the cytoplasmic inheritance. And next, there is a irregular segregation in the biparental inheritance. That means only from the female the traits are getting inherited. That's what it means. Next, it undergoes somatic segregation. What is the somatic segregation? See, if you know the cell, the nucleus, out of this, tell me like whether the cell organelles that are present in the cytoplasm, they undergo meiosis or not. Do they undergo meiosis separately, where there is like a crossing over, there is a recombination event that is taking place in this case, or it is limited to the chromosomes or the genes that are present in the nucleus? Which one is true, whether it is for nucleus or it is also for the cytoplasmic genes? Okay, our question will be like cytoplasmic genes undergo meiotic segregation or meiosis. Okay, it is only for the nucleus. Okay, so that's the reason why we say about the somatic segregation. See, whatever mutation, for example, what this mean to say is, if this is the nucleus and these are the cell organelles, okay, and they have the DNA. For example, there is some mutation that is occurring. If there is a mutation and there is any change in the trait, there is any change in the trait, or there is any change in the like structure of a gene. So this change in the structure of gene, like how it is going to the next generation, is it because of uh, division, cell division, because of meiosis? No. How this chloroplast is separating? This is separating randomly by because of binary fission, right? It is simple binary fission where the single chloroplast is split into two halves. So randomly, whatever the things have been changed or the gene sequence have been changed, that will irrespective, like irregularly divide, and that's how the segregation is happening. And this, while during the mitosis, the event of mitosis, that will make, when the this nucleus will make the two different cells here, because of this reason they will form, they will go to the different cells. So that's why it is a somatic segregation. Just know this. Okay, nothing in detail. Next, association with the organelles. As I told you, whenever I talk about the cytoplasmic inheritance, it is a true association with the cytoplasmic genes, and where these genes are located, either in the chloroplast or in the mitochondria. Okay, and out of which, if in the question they are asking about like the male sterility is associated with which genes, you have to directly mark it as mitochondrial genes. Do not get confused. Okay, it's not just the cytoplasmic genes, it is specifically the mitochondrial genes to which this particular trait is linked with. Next, the mutogenesis. So there is a some peculiar character in case of mutogenesis for the cytoplasmic inheritance. Can anyone tell me what is it exactly? Based on the mutation, if we treat certain mutation or the some specific mutation, we will be able to decide whether the gene is located in the nucleus or in the cytoplasm. Which is it? Is that mutation which can be used once this mutation is treated, and if the mutation occurs, that then it is most likely that the gene which is mutated is present in the cytoplasm. Which is it? Yes, yes, very good. It is the ethidium bromide. So ethidium bromide, it mostly acts on a cytoplasm genes. This is one different in a character regarding the mutagenesis, which can be classed under cytoplasmic inheritance. So these are some of the very important characteristics that you have to remember. Okay, next, the cytoplasmic inheritance. Okay, this is done. History, like everything, you just have to study on your own. Nothing much to discuss. And I told you about this variegation, the white part and the green part, and this is the variegated part where on the green background you can see the white patches. So apart from this, I told you about the petite mutants in the yeast. So when it comes to the petite mutant, remember these three important points. See, again in the peptides, which are because of the mitochondrial inheritance, there are of three different types. What are the three different types? One is like neutral peptides, and the first one, like second one, is like nuclear peptides. The third one, it is the suppressive peptides or the suppressive mutants. So the third one is the one which is truly because of the mitochondrial genes, which are some effect. Other two, like neutral, they won't be having any effect on the progeny trait, and the nuclear peptides, it is because of the genes present in the what? As the name suggests, it is present in the nucleus. Okay, this is about the petite mutant. So if you have already gone through in detail from my classes, or like if you have gone it on your own, just try to recall it. So nothing much specific can be asked. Just know the examples and know about the three different categories. It is neutral, nuclear, and suppressive mutants. And neutral, there is no change in the phenotype of the progeny, but in the suppressive mutant, the petite would be dominant. If it is present, it won't allow the mitochondrial function to take place. That's the drawback of this petite. Petite is like the other name for small, where there is no proper growth of the mitochondria. Fine. Next, there is a one more important type in case of inheritance pattern. See, one we got to know about nuclear inheritance pattern. One we got to know about the cytoplasmic inheritance pattern. But other than this, we have the third type. Third type is nuclear cytoplasmic inheritance. So what is the example for this nuclear cytoplasmic inheritance? I have to give the two examples, two important and very common examples here. What is the example? Yes, quick. I think like it's already displayed on the board. One is like in the plants you can Agiop, Iozap variegation in case of maize. That is the one example. Here, this is the one example. Next, the other one is like CMS. How CMS? CMS plus the restorer gene. So which is potentially exploited in case of hybrid seed production. That means I told the CMS is because of mitochondrial genes, but whatever action that is being taken by the genes that are present in the mitochondria, that can be abolished by the nuclear genes, which are called as restorer genes. There is some interaction between the genes of the nucleus and also with the genes of the nucleus with the genes of the cytoplasm, that is specifically mitochondria in the second case. These are the two important examples that you have to remember for nuclear cytoplasmic inheritance. Okay, molecular basis and everything you can skip it. Not the important one for now. If I say like it's not important, that doesn't mean that you should not read it. Okay, if you have read it for till now, it's fine. But if you have in case not read it, you can skip it. Now, this is the one another important point, like that is endosymbiont theory. So, what does this theory and how it is related to this mitochondria and the chloroplast? See, if you just break this term, what is this term says? Like endo is like within staying as symbiont, that is closely associated with the host cell. So, if you consider this is the nucleus, and you can imagine that the presence of the cell organelles, the characteristics of the cell organelles, for example DNA, let's take the DNA of nucleus and the cell organelles. First thing, the DNA, if I talk about, they are linear or circular in the eukaryotes. It is mostly linear, but the DNA of the cell organelles, it is circular. And if I talk about the ribosomes are what, like what is the ribosomes sedimentation coefficient in case of eukaryotes, and what is it in the prokaryotes? Is it same or different? Yes, quick. Is ATS in the eukaryotes? I am asking like ATS in case of nuclear. So the ribosomes which are present here will be like not in the nucleus. The ribosomes which are present here, like in the eukaryotes, it is ATS. But what about the one which are in the cell organelles? Whether it is ATS or 70S? Yes, it is 70S. So these two characters, just consider these two characters. These are matching with the bacteria or the eukaryotic cell. Which one is right? These characters are matching with the bacterial cell or the eukaryotic cell. Yes, it is matching with the prokaryotic cell, which is the bacterial cell. So the theory states that the long ago, when the evolution was occurring, the beginning of the evolution, the starting era, what happened? These primitive organisms have entered the host cell, which is obviously like eukaryotic cell. Then they made a symbiotic association here later, which evolved to be a part of the cell with some definite functions. What the endosymbiont theory means. And evidence for this theory is like the mitochondria and the chloroplast are of same size. One is size, which I did not talk about. The second one is circular genome, and the ribosomes, it is similar to the bacteria. The third one, the two I discussed. And next, the RNA polymerase. If you know about the RNA polymerase, the eukaryotes, the RNA polymerase is different, and the RNA polymerase in case of the chloroplast and the mitochondria are different from the eukaryotes, and it is similar to the prokaryotes or the bacteria. So, another evidence. And there is also the sequence similarity for some genes which are present in the CP and the mitochondria, which are actually similar to the that one of the purple synthetic bacteria. Like one good example is like rRNA. The rRNA of the mitochondria and the purple photosynthetic bacteria, they are similar. Based on this observation, this endosymbiotic theory is proposed. So any question, if it is asked regarding this theory, you should be able to answer it. Okay. Next, the features. The features of this organellar genes or genomes, it is also important. You can directly get this point in the exam. Make sure you read all these points once and try to understand each one of them and remember them before going into the exams. That's very important. I have almost discussed all of this in detail. Okay, so that completes your cytoplasmic inheritance. Next, it is like multiple alleles. Okay, So this is another very interesting topic, which is like one of my favorite topic from the genetics. Because the favorite, why it is, because I did not understand while I gave the ICAR PG exam back in 2020. I simply mugged up and gave this exam. Okay, mainly like Benzer experiment. I did not get the experiment. I simply mugged up whatever I used to know, like I just concentrated on cystron, Piton and Recon. Did not worry about the experiment, and I went for the exam. Even for now, like if you are not aware of the experiment, it's not a problem. You can give the exam and not many questions will be asked. But still, this topic is interesting. So let's try to simplify it. First thing, what are multiple traits, multiple alleles? The multiple alleles are like alleles. First thing is they are alleles of same gene. Okay, the first one, you have to make sure that you know this, that they are alleles of same genes. They are not the alleles of different genes. If I talk about A is a gene. That means if I say that A gene has a multiple alleles, I'm talking that it has more than two alleles. Then it can be noted as A1, A2, A3 or any number. Okay, that's how it is. And what is the realistic example? You can take the ABO blood group. The ABO blood group is a peculiar character. Okay, it's a one specific character, right? The ABO blood group, whether the phenotype, if I talk about, the person can have the A or B or O or AB, right? These are the possible cases. Fine. So now for these traits, what are the possible alleles? So possible alleles for these traits include IA, and IB, and these are the three. There are the three possible alleles for the four different phenotypic classes. Okay, so this is the one peculiar or the one perfect example for the multiple alleles. But other than that, please try to remember all the other examples that you have here. Okay, that are listed here. The RH blood groups, and there are several different types of blood group, but we only mainly study about ABA. There is also like LM blood group, MN blood group, sorry, MN blood group. So this is also there. There are several types. So these are also those are also the examples of the multiple alleles. And the human hemoglobin. So this is also example, self incompatibility in the plants. If you have studied, you have S1, S2, S3. These are also the alleles of a single gene. And next, the fur color in rabbit, which we discussed. C for coat color in rabbit, or the fur color in rabbit, where this is C plus is wild, and CCH is chinchilla, and CH is Himalayan, and C is albino, right? And in case of the white eye gene, the most studied trait or the most studied character for the field of genetics, which also has like white eye, it has Lozenzi gene, it has like a color rosy, and the wild type. So there are several mutants or the several alleles for the single trait or single character. Here, the example in case of Drosophila, and the notch brings in Drosophila, which is the dominant character, and the waxy gene. The mutants, so in these mutants, we can find there are several multiple alleles for a single gene. Okay, so this is the concept of multiple alleles, where these are the alleles of a single gene, but they are more than two. Now, what are the different types that you get in the multiple alleles? See, the multiple alleles can be actually divided into two different types, which may not be present in your BDC. Plus, if you are able to understand it, try to understand. One is true multiple alleles, and other one is the pseudo alleles. Pseudo alleles, what is the actual difference between the pseudo alleles and the true multiple alleles? If you want to know, so the difference is the alleles which we are talking about, they are from the different genes. Okay, the two alleles, if I give you two alleles, just for example, then if I say that these two alleles are pseudo alleles, that means the two alleles belongs to the different gene. One can be of A, one can be of B. But when I give you two alleles and say that they are true multiple alleles, that means both of them are belonging to the single gene. Okay, let's assume it as 1, 2, and this here 1, 2. They are belonging to the same gene here in the first case. That means they are true multiple alleles. But if I say that 1 and 2 are pseudo alleles, that means one belongs to one gene and other belongs to the other gene. But how to know? How to know this, whether the alleles belong to the same gene or no, not, because they behave like similarly. That's how like we can't able to differentiate them based on the phenotypic observation. So how we can do it? Yes, we can do it with the phenotypic observation, but it requires some test. For this one, for a very first time, the one test that was introduced, it is like recombination test, where when the mutants are crossed, like M1 and M2, if the progeny, like if the wild type is found, then this is considered as pseudo allele. And in the second case, when the two mutants are crossed, if this will give only mutants, then it is considered as the true multiple allele. But this is on what basis? This is on the recombination. Considering the fact that if this is the A homologous chromosome and this is like another its homologous chromosome, in this case it was assumed that if there is a single gene, there is no crossing over within a gene. Based on this assumption, this was stated. Okay, so the first thing, first test that came into existence was the recombination test. But what happened later? So I'll tell you what happened later. So before that, if I want to know about more about this ABO blood group, see, this is the A group, B group, AB and O. Okay, now if you know what are these red dots here, these are the antigens. So remember, if I say that the A blood group, what does it mean? A blood group means the person as the A antigen. If the person as a A antigen, then the person body, will it prepare the antibody for A or not? The same thing which I discussed in the previous class of erythroblastosis petalis. So tell me, like, whether it will prepare the antibody for A or it will prepare the antibody for B. Which one is true among these two? Yes, quick. So antibody for B. So that's what we have to remember always. For own antigen, the body will not prepare the antibody. Only for the foreign antigen, which is not present in the one body, like person body, the antibody is prepared. So now, if in the exam they are asking the like blood group A as an antibody for, you have to remember that it is will be B. If they ask for B, it will be A. But what if the blood group is AB? Whether they will prepare the antibody or not, they won't be having any antibody because both the antigens are present. If it is O, then the antibody will be prepared for both A and B. That's what you have to remember. And the allelic or the genotypic notation here, IA will give A, IB will give AB, IA and I will give only A, IB and IB is B again, IB and I is B, and small I is like O with a no antigen. O is nothing but no antigen, and AB is nothing but having both A antigen and the B antigen, and A is having A antigen and B is having the B antigen. That's it. Okay, you have to remember this. Any type of questions, either like father is having a blood group A, the mother is having a blood group B, what are the possible combination that are possible in the progeny? If they are asking, then you have to make all the cross combinations and try to answer this question. Okay, they will be very simple. You have to make sure you should answer that without fail. One thing I told you about is like true multiple alleles and the pseudoalleles. I'll come to this now. So I talked about the recombination test. So before that, let's understand the classical concept. So according to the classical concept, what was the understanding about the gene? See, what was our understanding about the gene was like, we made assumption that the understanding was the multiple alleles are mapped at the same locus. That means if this is a gene and if this particular A gene has A1, A2, A3, all of these will be at a same locus. If it is some place on a chromosome, and all of these places very close to each other, all of the multiple alleles will be present. Even this is true till now. And next, the no crossing over between the members of the multiple alleles. That means the A1, A2 or A3, there would be no crossing over between these two. If this is homologous chromosome, if it is A1 and A2, which are actually the genes of a same gene which is A, it was assumed that there is no crossing over between the multiple allele series. Next, the wild type alleles of a multiple allele series are always dominant. That was the other thing that was according to the classical concepts. And next, the last point, the multiple alleles do not show complementation. These are the four important points that were known from the classical concepts. But based on the understanding, deeper understanding about the gene, these concepts have changed gradually, that I'll discuss. First thing was recombination test. Why the recombination test? Like, you have to at least know why this was introduced. Remember, the recombination test was introduced to differentiate between what? Differentiate between whether the genes or whether the alleles are true multiple alleles. Again, what I mean to say, if I say that it is true multiple alleles, I mean to say that alleles belong to the same gene, or if it is pseudo alleles. Okay, so this was a one thing. This is why this was introduced, but it failed because of the reason that it was based on the assumption that the mutants, like if it is M1 and M2, if they belong to the same gene, A gene, so it was assumed that M1 and M2 will never undergo recombination or crossing over. But this was disproved by one scientist. Can you name him? Who disproved? Like there is a intragenic recombination. The term is like intragenic recombination. Who did it? Quick. Intragenic recombination. Yes, it's a Oliver. So this is what you have to remember. Since like Oliver provided the evidence in the which gene, it is one the Lucenzi gene, that yes, no, the crossing over can even occur in case of the multiple alleles of a single gene. So because of this reason, the recombination test failed to differentiate between the true multiple alleles and the pseudoalleles. And next, it was by E.B. Lewis who introduced the complementation test, which is till today accepted, and that's what we read. Okay, to differentiate the multiple like pseudoalleles and the multiple alleles. So what according to the complementation test? See here in the complementation test, you know about the I discussed about the cis and trans in one of the like in the previous marathon class. Now I told you like there are two different types. One is cis and trans. So based on the difference between the trans heterozygotes, the complementation test works. See, there are total of four different classes here, 1, 2, and 3. Now, if you know the complementation test works with the trans heterozygotes. Why only with the trans heterozygotes? Why can't we do it with the cis heterozygotes? What I mean to say is, why can't we use cis heterozygotes where the mutants are present in a same gene, same chromosomes, and the wild types are present in the opposite chromosomes? Why can't we use this combination to detect the true multiple alleles and the pseudoalleles? What is the reason for it? Why can't we do it? Because you can see, even if the two mutants are present in the same gene, or let's assume that two mutants are present in a different gene, but in the both the cases, if we are taking the combination of cis heterozygotes, there is no difference in case of phenotype that is produced at the end. If we can clearly see here, both cis heterozygotes, when we assume they are present in the same gene and they are present in a different gene, both of them give the wild type, that is active peptide. That's why we can't differentiate it. But why we are always using the trans heterozygotes? Because if in case the two mutants are present in the same gene, then in this case the mutant character will be only shown, and if it is present in the two different genes in the heterozygotes, trans heterozygotes, the active or the wild type is produced. Or in the simple words, remember complementation test, if in the like it will be definitely asked in the exams, please remember that when mutants show the wild type in the progeny, that means it is pseudoalleles. That means the two mutants are present in the different genes. Or in the question, if they ask two mutants are showing complementation, or two mutants are showing the wild type, or it's showing complementation, what does it mean? It means that two mutants are present in the different gene. And the statement, if it is like two mutants are showing or not showing complementation, that means the mutants are present in the same gene. That's what you have to remember. Okay, and the structural alleles and functional alleles, I won't be discussing in it now, it might be confusing people, so you can skip it. Next, so based on all this improvement, there was a changing concept of a gene, and the most important contribution for the changing concept of a gene was because of its one important experiment, which is Benzer experiment. So if in a simple words, if I have to explain what he has done the fine structure analysis of R2 locus in the bacteriophage. Okay, so which bacteriophage? Is it the T2 bacteriophage or the T4 bacteriophage? This is also confusing. Like if in the question they ask you both like T2 and T4, like which one will you choose? Yes, remember it is T4 bacteriophage. Okay. Now, so in this analysis, what he has done, he has mapped all the different mutants of the R2 locus, the mutants that are present in the R2 locus. Yes, try to map all the mutants here. Okay, there were 3000 mutants that were present in the R2 locus, and he tried to map it. That means try to identify the location of the mutants on a gene, sorry, on a chromosomes. That's what he has done. That's what it means, like fine structure analysis. So based on this, he has introduced three important terms. The terms are cistron, recon, and muton. So what are these? Cistron is nothing but the functional unit. If this is the chromosome, the part or the location where the gene sequence are present in terms of what? Nucleotides or nitrogen bases. So this is called as cistron, where the functional part of a gene is referred to as cistron. But what is recon? Recon is a nucleotide base which is indivisible by recombination. So what will be the number of nuclear nitrogen bases which can be which will be indivisible by the recombination, whether it is two nucleotides or the one nucleotide, whether it will be one nucleotide or two nucleotide, which cannot be differentiated because of recombination, where it cannot be divided because of recombination. It one nucleotide or two nucleotide is actually how, like if it is two nucleotide, for example 80, and it is like corresponding 80, can't be the recombination takes place between the two nucleotides? It's not possible. It is. So that's why the smallest unit which we are talking about, it is like one nitrogen base or the one nucleotide sequence. Okay, and this is about the recon. And I understand, like in the different books the meaning might be a bit different, but it is indivisible by the recombination, remember it. And muton, what is muton? The muton is a unit or in a gene which can undergo mutation. So now tell me, what will be the nitrogen base number? So how many minimum nitrogen bases or the nucleotide sequence is required to undergo mutation? What is the minimum requirement? Yes, it is again 1. So for both recon and muton, it is 1. So again in the exams, they are asking about the smallest unit of a gene. So there will be always some confusion whether it is recon or muton. So they won't try to confuse you by asking such questions. But in case if they ask you, just go with the muton. That's it. Okay. Don't apply anything, any concepts here. Forget all the concepts and go for the muton. That's it. Don't try to complicate yourself. That's it. This is all about the multiple alleles and the cytoplasmic inheritance, which I wanted to discuss. So this finishes the transmission genetics. I should have done it yesterday only, but I was exhausted. That's why. And I also wanted to complete it for one time, that nothing should be left behind. So I came up with this today's sessions and try to complete the transmission genetics in detail. That anytime you watch this video, it should be always feel complete. Okay, that's it from today's video. I hope like whoever is watching this video, like there would be some benefit for you people who for revising the content from these last minute videos. We are making our 100% efforts so that every bit of every moment, every second that we are giving, that should be useful for you. Like we have come up with the plant breeding revision series. We have come up with the most expected questions for the plant breeding. Then we are regularly uploading the plant pathology like diseases part, the 15 days challenge. They are regularly coming on the YouTube, and I'm discussing about the genetics parts, and most importantly all your doubts, all the mock test doubts that you are reporting in the mail. So you are getting the answers for the reporting, plus you are also getting the answers in the WhatsApp group also. But I would suggest, like what I would suggest in the last 3 to 4 days. So I don't know like how many of you are planning to attend the mock test and how many of you have done it. If you have not yet done it, so please try to attend at least one or two full length mock tests till now. Do not keep all the mock tests till the end of the day. Do not think that okay, I'll read till 30, I'll read for till 1 and I'll attend the mock test by 2. Don't do it. Every day, irrespective of like how much you have prepared, just go for the mock test. And remember, even till the exam day, even if the exam happens like 1 year after, there won't be like 100% satisfaction to any of you, including me. Okay, there won't be like 100% satisfaction that you feel okay, I've read everything is crystal clear, I'm 100% confident, I'll go and write the exams. This mindset rarely comes. there will be always extra pressure, there will be always something that you will not be confident about, there will be always some topics that you will repeatedly forget, you won't be able to remember even if you read the 100 times. And trust me, this is normal. Okay, this is normal. This happens with everyone. If you ask me, like I secured all India second rank in my PhD, but till the last moment I entered the all, I was very much worried, like whether I'll get a seat or not, whether I'll get a seat or not, whether I will get a seat. Forget about second rank. I was just wanting for a one seat. That's how the mindset will be. Whether you secure the all India rank 1, 2, or you rank secure like 50th rank, everyone's mindset is same. Only difference that can be made, it is through the confidence that you have and the peace of mind that you keep in the exam. So in the last 3-4 days, don't like blunder, make the blunder of mistakes. Don't mix all the content, the resources, read from here, read from there, like try to check the questions in the group, WhatsApp group, try to find the answers for whatever questions you are seeing in the group, and discussing the wrong things in with your friends. Don't do such mistakes in the last 3 to 4 days. No matter like whether you can revise like many things or not, that doesn't matter, but whatever you revise, that should be in your mind. Okay, I agree that in last 2 3 4 days, even if you have not revised everything, you can't do it. Agreed. Okay, but whatever you revise, if you revise genetics, if you revise like plant breeding, if you revise just CTech or microbiology or plant pathology, do it with 100%, so that in the exam all you should not feel that okay, this is pending, this is pending, this is pending. Whatever is pending, leave it. Not to worry about it. So for the 120 questions, you make sure that whatever you know, you will try to answer based on the understanding. That's it. Do not worry about the results. Do not worry about whether you'll get the MSc or not. Do not worry that okay, I should have started early. I was a bit late. I did not know about the platform early. Don't have any regrets right now. Okay. At this last moment, nothing matters. Only thing that matters is the confidence and how you are planning for the last 4 days, and how, what kind of, like whether you will have the peace of mind during the exam day or not. Have it. Just give it the best shot, get a best result. Not worry about it right now. So anything else? Anything? See, one thing you asked for the molecular genetics, I said yes. The second thing you asked for the fungal classification, I said yes. Third thing you asked for the marathon series for the plant pathology, I said yes. uh, for everything that you people are asking, we are saying yes. I ask only one thing, you should also say yes, which is like, share and comment on this video and support the platform, so that in the coming days, once you land into MSc, you will be going for the PhD. At least some of you might be really going into the PhD. Then by the time you come into the PhD, already the courses are available for genetics and plant breeding, seed science. But we will make sure for plant pathology, for microbiology, for all other subjects, including the plant science, we will make sure to have the fully fledged quality content for you people ready once you are planning for the PhD. Okay, and also for all other agriculture exams. It all depends like on your support. Anything else to be discussed? So I hope like the things that I have discussed till now from the transmission genetics is very much clear. If everything is clear, put some heart and show some love. Yes, quick. Okay, thank you so much. Thank you so much for watching this video. Revise well, have a good sleep, and we'll meet tomorrow or day after tomorrow. We will intimate this in the YouTube channel and also in the WhatsApp groups, WhatsApp channel. So if you have not joined any of our channel, the links are available in the description box. The WhatsApp channel link, Telegram channel link, the YouTube channel link, and the Instagram. Everything is available. Make sure to get the right updates. Prepare well for this exam. Thank you so much. Good night.